I am Krishna Gopal, presenting constructions of DNA codes with various constraints, work jointly done with Professor Trish Banerjee, Department of Electrical Engineering, IIT Kanpur. Data storage is one of the burning issue and researchers are looking for possible solutions for it. DNA-based data storage is one of the smart solutions to store huge amount of data in a small place. DNA is the sequence of four nucleotides: adenine, thymine, cytosine, and guanine. These nucleotides are denoted by A, T, C, and G respectively. A and T are complement to each other, and similarly, G and C are also complement to each other. For a given DNA sequence, the reverse DNA sequence is the DNA sequence in reverse order, and the reverse complement DNA sequence is a sequence of complements in reverse order. A single-strand DNA sequence folds upon itself and forms secondary structures by pairing between A and T, between G and C, and between G and T. While reading and writing such DNA, we have to unfold it, and therefore we need additional resources. A DNA sequence has a home polymer of run length L if all the nucleotides are the same at some L consecutive positions. For example, consider a DNA sequence AG, TT, AC, CC, CCT of length 11. It has Cs at third and fourth position. So it has homopolymer of length 2. The melting temperature of a DNA sequence depends on the GC weight of the DNA sequence. The GC weight or GC content of a DNA sequence is the total number of Cs and Gs in the DNA sequence. In this example, the GC weight of the DNA sequence of length n is 5. While reading and writing the DNA sequence, insertion, deletion, and substitution errors are possible. Example of these errors are illustrated over here. In this poster, we are limited to substitution errors only. And an MDH DNA code is the set of mDNA sequences, each of length n, where DH is the minimum Hamming distance of the code. An example of 483 DNA code is illustrated here. By reading and writing a DNA sequence from an NMDH DNA code, we are interested in following constraints. Hamming constraint. In the Hamming constraint, the Hamming distance between X and Y is at least DH. Reverse constraint. The Hamming distance between sequence X and the reverse DNA sequence YR is at least DH. Similarly, for reverse complement constraints, the Hamming distance between X and YRC is at least DH. If each DNA code word has the GC weight W, then the DNA code satisfies fixed GC content constraint with weight W. If W is the half of the sequence length, then it is called GC content constraint only. Similarly, if each code word of the DNA code is free from home polymer of run length of more than L, then the DNA code holds L home polymer free constraint. And if each DNA code word is free from secondary structure of stem length more than R, then the DNA code satisfies R-free secondary structure constraint. In the work published in IEEE Communication Letter 2021, we have constructed home polymer and secondary structure avoiding DNA codes with Hamming and reverse complement constraints. The parameters and other details are given here. Now, in the paper accepted in ISIT 2022, we have constructed home polymer of secondary structure avoiding DNA codes with reverse complement, Hamming, and GC content constraints simultaneously. The parameters of the DNA codes are mentioned here. Similarly, in the poster presented in Stanford Compression Workshop 2021, we have presented a family of DNA codes that avoid secondary structures and satisfies Hamming constraints and reverse complement constraints. In this table, we have compared properties of DNA codes with the code available in the literature. Here we obtain that DNA codes with code rate 0.514, relative minimum Hamming distance 0.333, and it satisfies reverse complement and GC content constraints. Also, the DNA code avoids homopolymer and secondary structures. Milinkovic and Kashyap obtained DNA code with code rate 0.401 and relative minimum Hamming distance 0.571 with a GC content constraint. Also, the code avoids homopolymers and secondary structures. Thank you for watching the video.